this computer because they told us don't use the cloud. So I will do my best to download these and put them in Blackboard in the unit itself. So if you've gone to Blackboard, you see that we're going to do it by Unit 1, and I will just add lectures, Tuesday, Thursday lectures with the notes. Everything that I passed out to you will be there. If there's any study guides, they'll be put in there. Okay, and that's where your quizzes will go to. Okay, so everything will be in your Unit 1 up to Exam 1, and so that's where that'll be found if you're not real familiar with Blackboard. So the video has started, so now I need to go to share screen, and we're going to share our PowerPoint screen, and we're going to get going. I think. Here we go. Okay, Randy and Rachel, does everything look good? See the PowerPoint? Good. And I tell you, if you're on Zoom during lecture, I will be calling on you just as I would anybody in the classroom too. Make sure you're still there if you're not going to use your camera. All right. I like to follow in my notes, so you'll see me holding on to my notes as we go through, just so we can point out things that are important. And like I said, this is a microbial physiology and metabolism kind of a review in one lecture. And it sets us up for what we're going to do downstairs at starting at 12:30, which is gram staining. Now you have a gram staining exercise. I set up some cultures yesterday. They're numbered 1 through 10. Each table that comes in will have you'll have 2 plates, 1 for each of you as a pair. And you'll get your gram staining going. You'll also need your microscope. So there will be a sign-up sheet. Remember that I want you to claim 1 scope for the entire semester. So that means scope for this class, and this means a scope for the same scope for hematology, the same scope for immunology if you're in there, and the same scope for Heme 2 for those that are in Heme 2 in the room. Okay? We got enough scopes that we don't need you swapping in and out each class. You come in, you grab somebody just because they're not in there. Because they want to know that they're not having a scope that you're using while they're not using it. And come in and maybe you cleaned it, maybe you didn't clean it, maybe you messed it up, maybe you just, we've got enough. Because I did a visit to another program out in Seattle area. I used to go inspect other programs and be their site visitors when they did their accreditation. And one of the things that struck me then was the students, we got to get the students out in the room like this by themselves and said, okay, now tell us the real stuff. What goes on? And they said, well, we need equipment. So because what we do is after class, we actually run to the lab and grab the best scopes. Because there's only like 4 really good scopes. And then we let everybody else struggle to use the others. So I was like, oh my gosh. So I went to the dean, and the dean said, what's something we can do? We can do anything you need us to do. And I said, well, work on your equipment. Your students are running a sprint to try to get the best equipment for the day, because they know if they don't, they're going to get something terrible. So go ahead and find you a scope. If it's not perfect, we'll work it out. We'll clean it. We'll get everything going on it. And if we can't, then we'll swap out and get you a different scope that somebody else hasn't claimed. But there's 25 of you here, so we've got 40 scopes. Okay, we have 20 in each room, and it doesn't matter. You're going to be using it back and forth. You can cross back and forth and get your scopes when needed. But we're going to need it today. This might be one of the few days we need it in micro, but today is the day, because we're going to look at the gram stain slides that you make. After we do the gram stain slides, then we're going to move to set up for next Tuesday, which is you're going to take the plates that you just gram stained, and then you're going to streak to a new plate. Okay, so you're going to do a streak for isolation. We'll just let it grow and grow and grow over the weekend. I'll be back up here, so if the growth gets to a point where we can pull it out and just put it on the countertop, we don't have to incubate it the whole time. and you can see how you did on your isolation on Tuesday before we get started on staff. Okay? We kind of missed the lab on the schedule, so we're kind of pushing the labs into Thursday. That's on your schedule. And then we'll be caught up for next week with staff to start on Tuesday and read staff on Thursday and keep going on schedule. So I think we've done good with that. So some of the things that we're going to look at is morphology today. And basically size of the bacteria. Of course, we know these are very, very small because we're going to put them on a slide and we're going to stain them. And we're going to see thousands of colonies today, right? We're going to see an individual, right, a cocci of an organism today. We're going to see something else today too. So we have 10 places to look at. And why I'm putting you in pairs is because, and we're going to have to move that Zoom window, aren't we? That's going to be interfering. What do you have? Let's see. Let me try that. There we go. How about that? We'll try that for location, see if that works. So in pairs today downstairs, you're going to do yours, and then you're going to share slides, not microscopes, okay? Slide's going to come across to you of the other one. So they're set up where there's going to be different ones. You're going to have a different one from your partner, and you're going to need to see the other one to see the distinct. So you can probably figure out one's going to be gram positive and one's going to be gram negative of the pair that you have on your table. I don't think I'm giving away much there. But. So a human hair diameter, so if we pulled a hair out and the diameter is not the length, the diameter is, you know, from one side to the other of the stalk of the hair, you see that it's about 17 to 180 micrometers. And you can see that mycoplasma and rickettsia. Now, when I run through these names, today it's okay. We're just going to introduce names. So don't raise your hand and ask me what that is. Okay, you have a textbook. You can start looking these up. But we're going to cover each one as we move through. So right now we're just kind of introducing lingo. So don't panic. Don't get all, oh my gosh, I don't know what in the world that is that he's talking about. We're going to throw in a lot of examples that we're going to revisit in a whole lecture coming up down the line. So when we're talking about mycoplasma, rickettsia, E. coli, bacillus, borrelia, we're going to see all those again. Okay, these are organisms you're going to need to be familiar with by the time we get the semester over with. And I don't know if intro micro had you familiar with these names or not. Don't know, right? Some of you will say yes. Some of you will say no. I wasn't there, so I don't really know what they're teaching. But what we see is different sizes. So you may see something on a gram stain that's obvious, like, oh gosh, I could tell that was a cocci. Then you may have another organism on a gram stain. You're like, I need an electron microscope. I need to see this closer. Can I get a better magnification? It's too small. Can't really tell. That's, you know, is that a small rod or is that a small cocci? I don't know. Right? Some of them are that hard. So 1,000 times magnification, maybe not enough for you to obviously tell what you're looking at on a gram stain. But most of them are going to be pretty big. And what you'll notice on our PowerPoints is we'll fill in the blanks. They're in red to help you out, all right, to keep your attention. We have little red things that we leave out of the PowerPoint, but it's in your notes. I mean, it's on your PowerPoint, but it's not in your notes, so it's kind of weird. But we'll do a 2 to 4 micrometers of E. coli. Bacillus is bigger, right? So it's more obvious when you look at it. You're like, I'm not going to mess that one. There may be some that are really, really small, right? 0.3 to 0.4 for mycoplasma and rickettsia. Then we have our morphology, which is the shape. So we have cocci, which are round. We have bacilli that are rod-shaped. So you want to say it looks like a rod, it looks like a cigar, whatever you want to put in your head. We have coccobacilli, which are elongated cocci. So some of you will look at gram stains and go, is that coccobacilli or is that actually a rod? Okay, and we'll see the distinction. Usually what you can tell is that it looks like two cocci together, but it's not diplococci, it's just kind of an extended cocci. We actually have some that are way different in shape, which are helical, okay, which look like a spiral in the old spiral notebooks. I know y'all haven't seen, well, there's one. How you have a spiral notebook. Show them what a spiral looks like. Yeah, see that end of that? That's a spiral. So we have some bacteria that shape that way, okay? So we have some different shapes, and we'll see a little diagram here in a minute. And your book has this too. They can be arranged different, right? We know that when we get ready to look at staph and strep in the next couple of weeks, we know staph is a cluster. So the cocci of staph are going to be grouped together like a group of grapes, they say. You know, if you hold grapes up, you see all these little round grapes all together, touching each other. And that's what staph does. Staphylococcus does that. Strep, on the other hand, goes in chains. So you'll see a chain of cocci. Okay, so we can tell that on a gram stain. As soon as we get a gram stain of strep, when we get a gram stain of staph, we can definitely tell who's who. We have them in pairs. We have diplococci. It's very, when diplos are all over your slide, that's great. Okay, but a lot of times diplo, if you're really aggressive on mounting your organism on your slide and you like to really rough smear it on there, you may scatter those diplos where it looks like a lot of single cocci. Okay, you've broken them apart. But if you look real close, you may see two cocci together, two diplos, two cocci touching, and you want to go with the majority. So you'll look at it and say, well, there's a diplo, diplo, diplo. I'm just seeing diplo, so I'm thinking diplococci. Strep we just talked about with chains, okay? We have gram-negative enterics. That's what we'll call these. The enterics are like GI organisms. You know, our E. coli is our main one there. Staphylococci and diphtherioids can occur in packets, palisades, okay? So we got some different organizations of what we're looking for. So when we do a description of the bacteria, this is going to be part of it. You know, when we talk about the midterm coming up, you know, when I said, hey, I'm going to give you a description of the organism, well, this is going to be part of it. We'll say, present as gram-positive cocci in chains, right? Or we'll say gram-negative rods, stuff like that. So these descriptions are very helpful at the beginning of the story, of being obvious of what we're looking for or what we might see on the slide. So we definitely have our coccus, our diplococcus, our diplo-encapsulated cocci. That may start to kind of look like a coccobacillus a little bit. We get our strep in chains, we get our staph in our cluster of grapes. Okay, we might have some over here, like a helical form, Helicobacter pylori. Does anybody know what that one causes? Just off, I'm just kind of seeing where you stand in your knowledge. H. pylori. What's that?
So we had that in parasite.
You had that in parasite? Maybe something similar in name. This is the bacteria.
Yeah, there was a pylori then?
Mm-hmm.
But not H.
Not helico. H. pylori is the bacteria that causes ulcerations in your stomach lining. So we see that doctors will order H. pylori antigen test, okay, if somebody's having abdominal pain for a long period of time and they suspect that that's the bacteria that's found, and we treat it, right? They treat it now with antibiotics. So back in the day, my dad would have like, oh, I had a bleeding ulcer one time. I won't tell you about it. I'm like, thanks, Dad. But what he was saying, oh, they're going to take my whole stomach out. The doctor wanted to just cut me open and take it out and take that part, and I wasn't going to have a stomach. Well, today, if you have an ulcer, usually the cause of it is H. pylori, the bacterium down in your stomach, okay? And you take an antibiotic to what? Knock it down, okay? So it keeps you from irritating the stomach lining. We'll get back to that, don't worry. We have the corkscrew, Borrelia burgdorferi, down here. one of my favorites. And that's the Lyme disease. I don't know if you've ever heard of Lyme disease, but that's the bacteria that the tick has when the tick bites you and puts that in tick bite, and you end up with a Borrelia burgdorferi infection that turns into a Lyme disease. All right. So this is definitely a great little guide. You'll see this on Facebook and stuff. People share it. Here's some more. We see our Staph aureus up here. This is our MRSA, wound infections. You'll see a lot of that. Here's our strep pyogenes. This is our strep throat. Here is our Neisseria gonorrhea. Bacillus anthracis, anthrax. I don't know if y'all probably weren't around in 2001, right? Were you? 2001. Right. You know, after that, and it's still happening today. Somebody gets a package in the mail, a hate crime. Somebody might put some anthrax in an envelope and send it to somebody. E. coli, this is our #1 urinary tract infection organism. Naturally occurs in our bowels and our stools, and it ends up in a urinary tract when it shouldn't be, and it causes a UTI. Vibrio cholera. So we have different shapes, and we're not going to get into the cyanobacteria down there. That's environmental. So if you've been in environmental science, you might have seen those naturally occurring in a pond and turning it colors. Okay, there's some really neat pictures this summer, I think, of somewhere in the oceans where they had cyanobacterium turning the water like glowing in color at night. Our bacteria also have capsules and slime coverings. And those that are in immunology, which is most of you, or you've already had it, this is kind of like the part of immunology that the bacteria have. They evade our immune system because of these capsules and slime that they have, and make it harder for the antigen to be detected. Okay, so they cover it up. So the organism secretes it, it covers itself, it's loosely bound on the outside like a slime layer. And then if the material possesses structural organization tightly adheres, it's called a capsule. So you have a slime loose and a capsule tight. And then the chemical composition of the slime consists of polysaccharides, okay? So that's just sugar subunits. And the ones that produce this, we call them encapsulated. So that's another part of the description. Something like Klebsiella, we'll talk about it being encapsulated because when it grows on the plate, we see this big puffy capsule that comes up and covers it up. How we doing? Doing good? Nothing scaring you so far? I'm glad y'all came back for day 2. It's always a good sign. Makes me feel like I didn't do too much on day 1 to scare you out of here. Capsules and slimes. This material, like we just said, inhibits the phagocytosis of the white blood cell. Okay, so it is therefore a virulence factor. It makes this organism more virulent because if it can then avoid the immune system, it can then set up an infection and cause you to be sick. Okay. If it didn't have this, your immune system would probably attack it, destroy it, eat it, get rid of it before it had a chance to set up an infection. Capsules are very antigenic and invasive of the body. Encapsulated bacteria induces the production of antibodies. So we get antibodies going against that capsule. Okay. The antibodies can be used to identify the microbes that we get. So we do a lot of our testing, which we'll be doing, where we have antigenics determining antibodies, where we get an antibody to test to see if you have it. So with a bacteria infection, we can do some testing. So if we have a certain bacteria and we want to know if we have it, we can search and put it in with some antibody and see what happens. And if it binds together, we know you have it. If it doesn't, we say you don't. So we see that a lot in immunology, serology, and micro together. And then what we see is if you have enough infection, we get an antibody titer that lets us know that you have this. Now, for those in immunology, I think everybody's in immunology but a few. I think Preston was the only one that was in immunology yesterday, but not in micro.
I think Lauren might have a question.
Oh, okay. Lauren, raised your hand.
Well, actually, I'm just now joining the Zoom call because the email that was sent out, it wasn't working. So me and Kathy Grimm, I think she's still out there. I don't know how to invite her, though.
Okay. What do you mean it's not working?
The email that was sent out, we clicked on the email part, but it wasn't working. So I tried, we tried to email you, and then I looked around on Blackboard and clicked the one on the announcements, and that one's working. But
Okay.
Kathy is still in the other one, and I don't know how to invite her to it.
Okay. So you picked a different link?
It said the one for today, though, so I clicked that one through the email, and it wasn't working.
Take you to this?
It didn't take me. It did not. It took me to a different one, but it said it was for today. So.
Okay. I'm sorry. I don't know what happened. Were you in class earlier?
Yeah, but then I looked at my email and it said that I had to be on the Zoom, so I left. I thought I was gonna get in trouble.
No, Zoom is for the students that can't be here. So if you've been quarantined, isolated, then you come to class. Sorry, I don't know if I made that clear. But
Okay.
the reason we're Zooming, it's an option that we're providing for those that can't make it to lecture, which you could, for other reasons, if you don't want to come to lecture, you're definitely eligible to do it. But then we're recording it too. So I didn't know while Kathy, she might have just been doing that because she knew it was going to be recorded. I don't know.
Okay then. All right. Sorry.
Okay. But yeah, if you can get a message to her, that'd be great, and tell her, hey, sorry about the link. What I tried to do with the Zoom was I tried to make it recurring, and it wouldn't let me.
Yeah.
It had to be a daily class for me to make it recurring. But I was trying to make, like, this meeting's going to happen every day. So I've been just assigning a new link to this everyday meeting. And I'm sorry that the email had the old link in it, but I usually do it at the same time, which is I copy the invitation into the announcement, copy that same invitation into the email.
Okay, I'll try to reach out to Kathy.
Okay, yeah, I'll try to reach out and tell her I'm sorry. But it's being recorded, and I'll upload it for anything she feels like she's missed.
All right.
She can definitely go back and rewatch it.
Okay, thank you.
No problem. Thank you. Yeah, you're not going to get, if you're coming in and you're like, oh, he's going to Zoom, we're live. We're going to stay as live as we can be until they tell us otherwise. But we're going to do live lectures, and you can be in here, but if for some reason you can't make it, some reason but we had to provide the isolated and quarantined students an option, which was a hybridation of the class, which meant we're going to do this every day. As long as technology holds up, it's going to be great. So we're trying to stay face-to-face. But that being said, since it's been brought up now, if exam 1 doesn't go well, it's only going to be with Zoom invite to the people that have to be out. Okay. Right now, it's optional. If you don't feel comfortable being in class right now until you see what's going on with the virus, that's fine. That's your option. You can go home, watch it on Zoom, interact with us. But exam 1 is going to be the deterring factor. So if you're on Zoom half the time, because you just want to be, and then your exam's like a 70 when we get done, then that option's going to go away. It's just going to be, you're going to have an email sent to me after that says you've been isolated, quarantined or isolated for the next 10 days or 14 days. That makes sense? So I didn't want to come in here and not have anybody in here, but if you don't feel comfortable, by no means are we forcing you to come in here. Now, lab is required. There's no Zooming of lab. You need to be in lab. The ones that are missing lab right now, we're having to try to keep them informed and then kind of give them a quick, when they get back on campus, we'll get back into it a little bit, show what we did, but it's going to be hard if you miss that many weeks of lab. So lab attendance is still required, but right now lecture is optional. We've had some reach out and say, I just don't like being here right now. Is that okay? Yes, right now it can be okay. Is that okay with you guys? I mean, are y'all going to learn? Are y'all going to act like y'all going to get up and leave now and catch it on Zoom? I hope not, because it might not work. All right, let's get back. I think we can get through this lecture. All right, so we're going to do antibody titer. We're going to do that in immunology. Okay, so we're saying it loud right now because we're going to do an antibody titer in immunology, where we look at where we call something positive. And that's all that means is how much antibodies present. Is it enough, right? Because with what's going on with COVID right now, do I have enough antibody to give me protection, and how long is that protection? Is it for the rest of my life, or is it going to be like for next 3 weeks? Or, could I be infected in, say, January if it comes back, and what happens? All that good talk. So how do we determine that? We actually do an antibody titer, and we say at a certain level that we find, and this is just by dilution, okay, which we'll be doing in immunology too. We dilute down the sample until we quit seeing agglutination. Okay, so the antibody, if it goes away as soon as we dilute it one time, that's not enough. Okay, so we can gauge the amount of antibody there. That's a titer, the amount. And if it's at a certain level, okay, of dilution, then we consider it a positive antibody. And we call it negative antibody, even though it's kind of there and it's really there, but it's not enough to provide protection. Another one of those tests that we'll cover in immunology is called the Quellung Test. Quellung is employed to test clinical specimens, so kind of same kind of thing we're talking about. I don't want to get too much into immunology here, but you do need to know kind of this name. When you see it again, you kind of need to know it's a test used to test for the encapsulated bacteria like strep pneumo or Haemophilus influenza, okay, to see that it's there, and that reaction tells us if we have enough there. So the biggest thing about gram staining that we're going to be talking about is the cell wall, right? So all bacteria from your intro class, all bacteria have a cell wall, right? Makes them different from a eukaryote, which has a cell membrane only. Correct? That sound familiar? Prokaryotic organism, y'all understand that part? Okay, good. Cell wall is responsible for the shape. Bacteria that lack a cell wall, there are some that lack it, and they can swell if they're put into a hypotonic environment. Right? Hypotonic simply just means more water than stuff. I don't know if you've ever heard it put that way, but that's the way I like to describe it. Okay, an isotonic solution would be equal water and stuff, and that's what we see with saline. So when we do saline with blood bank coming up in the spring, you're going to hear a lot of talk about saline. And saline is added, it has salt and water, and it keeps the cells from doing what? Swelling or popping or crunching down and shrinking. Cell wall is also a major source of antigenic determinant, okay, to stimulate the production of the antibody. And then this antisera, okay, that we can use contains type-specific antibodies. And we use antisera, which is antibodies, to detect the cell wall, okay, antigens. And we have this identify pathogenic bacteria by using that serological test. And what does pathogenic bacteria mean? Somebody interact with me. I'm not gonna. Pathogenic.
It causes disease.
What's that?
It causes disease.
Disease, that's exactly right. Disease-causing bacteria. Was that through, is that Lauren? Thanks, Lauren. So we divide our bacteria into two major groups, gram positive, gram negative. You're going to start that today. And the basis of the gram stain is how we determine it. And gram positives have something that the gram negatives do not. Okay, they have what? What's the difference between the gram positive and the gram negative organism? Back to intro. Some of you said you did gram staining in intro. What do you remember about the difference between the two?
It has to do with the chemical reaction with the staining.
Staining, there's something that's really going to help hold the stain. Okay? It's this right here. The peptidoglycan layer in the gram positive organism is thicker. In the negative, it's very small. Okay? So the stain, that crystal violet, the primary stain that we use, gets that peptidoglycan layer stained, and it holds it. Okay? It resists the decolorization. Okay, so it stays purple. Another common term. The gram positive also is composed of this phosphorylated polymer known as teichoic acid. You see again the gram stain is these teichoic acids. They're linked to peptidoglycan layer. So we're going to see that. We're going to see a picture if you don't remember it from intro. I want to go, did y'all take intro? Was it just last spring or was it last fall? Everybody's had it, right? Trick question there. See anybody's hand go up. No, I never had it. All right. You might have gotten a pass into the admissions process then. The composition of the cell wall for gram stain gives them the capacity to resist this decolorization that we use with methanol downstairs this afternoon. And some of our bacteria like Mycobacterium, Actinomyces, and other fungi, yeast and molds, which we're going to get at the end of this semester, also stain as gram positive. So we may see some weird things staining gram positive depending on what we actually have growing on the plate. Because if you didn't know, yeast can grow on our plates. Okay? So if we pick up yeast, we can have that gram positive too. So here's a picture from your PowerPoint that shows this. So here's the cytoplasmic membrane, okay, and here's the peptidoglycan layer. You see here's the teichoic acid, okay, here's some more proteins associated. So this thickness here of the peptidoglycan layer determines whether we're going to have a gram positive or gram negative. So this is the gram positive, okay, so it has an inner cytoplasmic membrane, and then it has the peptidoglycan layer, and that's it. Gram negative, they do not hold on to the crystal violet because they're going to decolorize. So we're going to actually take the color away from a gram negative organism and have to counter stain it with our safran. Okay? But the reason is they must result of high lipid content in their outer envelope. So a gram negative organism has the cytoplasmic membrane, it has the peptidoglycan layer, and then it has an outer membrane above that. So almost like the stain can't get to the peptidoglycan layer, even though it's thinner. Okay? So it's very thin. If alcohol dissolves the lipid components of the cell wall, then whatever staining we had on the peptidoglycan layer, and then it leaves. Okay? And we'll see this today as we put the methanol on top of our organism and the crystal violet runs off our slide. We're going to see that decolorization. Cell walls are very porous, meaning the crystal violet is washed out. The high lipid content of the cell wall of a gram negative is also instrumental in preventing the uptake of the large molecules. So what happens is this outer membrane of the gram negative can also affect our antibiotics that we prescribe to the patient. Okay, like benzyl penicillin and actinomycin D. Okay. They're good examples of molecules that will not readily diffuse across the outer envelope of a gram negative. So you can see that maybe a gram negative organism might be a little bit penicillin resistant. Not that it did anything genetically to change itself, but it's just naturally built to what? Prevent penicillin from punching holes in the cell wall. So if you know much about cell biology, we have proteins inside the cell wall that are porins that let things, it's like a channel built into the wall, okay, that lets things move in and out. Okay, so a gram-negative cell wall may be one factor involved in antibiotic resistance to the beta-lactam antibiotics. So we see gram-negatives aren't like, oh, let's take penicillin and that'll take care of our urinary tract infection that's caused by E. coli. That's not going to work too well. But our strep throat, that is gram-positive, right? Take penicillin and boom, it's gone the next day. Okay, so penicillin's great for some things, but doesn't work on all things. And this is why. Okay? So here's our gram-negative bacterial cell wall. As I said, it's a double. We have an inner membrane. We have our peptidoglycan layer here, and we have our outer membrane. Here's the poron channel. Here's our polysaccharides taking shape outside of that outer membrane, which are good for antigenic determinants. Okay, and then we have our capsule, our lipopolysaccharide capsule out there that covers the entire organism. Okay, out here, it helps it evade the immune system. So here's the difference in the thickness of the peptidoglycan layer. You see it in blue compared to a gram-negative at the bottom. Okay. So a gram-positive is definitely going to take up that purple stain, and a gram-negative is going to lose it. So this is getting a little deep into what we're going to talk about later. But when we talk about Salmonella and Shigella, you know, we hear that all the time, right? There's always a Salmonella outbreak of some kind on our lettuce or our broccoli or anything that's grown outside that gets pooped on. It's a good way to think about it. Okay, and it gets brought into the restaurant or brought into the grocery store, and we eat that, then we could have a Salmonella outbreak. Okay, so one of the things that confers, there is a core polysaccharide to all members of the genus, but does not confirm specificity. There's a side chain that does, and the side chain is called an O, or somatic antigen, and it allows Salmonella and Shigella to be identified serologically. So we're going to do this later in the semester, where we actually look for the presence of that antigen. You got it. We're going to use an antibody specifically designed for that antigen to test to see if it's there. So we can actually grade out a Salmonella, whether it's, we got different types of Salmonella. Lipid A is another thing that's in the cell wall, another component of the lipopolysaccharide. It's highly toxic, okay? So if a bacteria possesses the lipid A, okay, it can give rise to an endotoxin, because this is on the gram-negative cell wall. This lipid A then can turn into something that causes sepsis. And sepsis is a condition, okay? So when we talk about, one thing that we do a lot in micro is we do blood cultures. Okay? We're responsible for the blood cultures that are drawn at the hospital. And most ER patients get blood cultures, okay, once or two times while they're there. And a lot of times at the beginning of my career, the blood culture was considered the sepsis indicator. Okay? Oh, they're not septic because his blood culture is still negative. Well, that's not really the case, okay? Sepsis is a condition of the patient not doing so well. Okay, their breathing gets shallow and low. They start running a temperature. Okay, their white count increases, those kind of things, and they get in trouble really quick. Okay, so a lot of times our ER physicians have a sepsis protocol that if somebody comes into the ER and they look at them and go, this patient's septic. Okay, they hadn't done a blood culture one, they hadn't done a temp on them, they just see them and say, let's run the sepsis protocol, which is I want 3 antibiotics given to them, I want 2 liters of fluid given to them, draw your blood cultures and go take them and see if it's in the blood yet. But I'm not going to wait on your blood culture results to call that patient septic. Okay? Sepsis is a definite, has a protocol to follow to call a patient that. So when I started, it was always like, we're not worried about that patient. They can't, or we get a positive blood culture and say, oh, that patient's septic. And they've already been treated beyond that. You know, they're not waiting 5 days on a blood culture result to treat a patient. So sepsis is an extremely dangerous condition, can be lethal to the patient. And what's been found is if you go with that protocol and give that patient those antibiotics, give them the liter of fluid, 2 liters of fluid right off the bat. This is like right when they come in the door. You start all that they survive whatever is going on with them because you treated the sepsis condition before you found out exactly what they all had, okay? So this DIC, and this is the question of the day. Have you ever heard of DIC? Anywhere? Huh?
On TV shows.
On TV show.
Like Grey's Anatomy.
Grey's Anatomy talked about DIC.
Probably.
So in Grey's Anatomy, let's follow up the Grey's Anatomy story. So the DIC patient, how did they find out they had it? Where were they when they found out? Because this is a shocker to the doctor when this happens, especially the surgeon. Remember?
Surgeon, so surgery.
It's in surgery, right? The patient is under DIC, which means they can't. It really sounds like, and this is another, you're going to get this in coagulation when you get there, okay? And I didn't know if we talked about it in principles yet. Coagulation, disseminated intravascular coagulation sounds like what? What's coagulation? Clotting, right? So it sounds like they got a clot. That's not the case. Disseminated intravascular coagulation means they've lost the ability to clot. They don't have any clotting factors left. So when you cut them on the operating table, they bleed and they don't stop. Okay, so they bleed to death on the operating table. That's what's important about doing our PT, INRs, and PTTs. Those are our tests that we test before somebody goes into surgery, so that we don't we don't get to this, right? This is not good. The doctor cuts somebody open and they can start bleeding out unstoppable. They're going to be pretty upset with the lab because you guys messed up on PT and they didn't have clotting factors before they went in there. You can give them all the plasma you want to and all the platelets you want to. They're going to die before it works. Okay. So disseminated intravascular coagulation is a condition that occurs with sepsis. So you can see if somebody came in the ER and they needed to go to surgery and you took them in there really quick because it's emergency and you didn't get those PT INRs done, and then they bleed out and it's not good just because of the sepsis condition. Both gram-negative and gram-positive bacteria possess peptidoglycan, glycopeptide. Okay, so they both have peptidoglycan. I'm not going to ask you to know the N-acetylglucosamine or the N-acetylmuramic acid, sorry. But if you want to know those, fine. Okay. So there's long chains of amino sugars. They're cross-linked. So basically what we're seeing is these are the ways of bacteria stay what? Structured. Right? They just talked about some bacteria don't have a cell wall, and those that don't, they lose their shape. Okay? This keeps the structure of the bacteria solid. It's like a framework. So we know some can exist without it. Okay? But they're real fragile. So you might find that they're hard to, what? Culture, if they're fragile. So here are some of the things that if you have a cell wall deficient bacteria, we call them protoplasts. All the cell walls removed, resulting in a spherical cell that's found only by the cell membrane. Protoplasts are derived from a gram-positive bacteria, so this is kind of like an outpocketing of the organism. Spiroblast is another group of cell wall deficient bacteria. Mycoplasma species that we see in the lab is a genus of naturally occurring cell wall deficient bacteria. They grow very slow. You have to put them in enriched hypertonic media for culture. So that's what I mean by they're hard to what? Culture. So Mycoplasmobacterium, we may need some other way of identifying it. We're almost through it. I know this is just great stuff. I know that. Because we're just talking about these cell walls, and now we want to talk about the cytoplasmic membrane. Okay, so you have the cell wall, and then you're going to have the cytoplasmic membrane, okay, that's got 70% protein and 30% phospholipids, and it's this bilayer, okay, and anytime I've done cell biology or cell anything, right, it's always what? The phospholipid bilayer. You heard that term before? The term of the hydrophilic head and the hydrophobic tails, which means the heads stay out here and the tails come together like this and the membrane looks like this as we come across. Have y'all seen this? And stuff can intertwine through there and stuff can go out of the cell and come back into the cytoplasm because it's semi-permeable, right? You heard that at all? Okay, good. Now you have, because we have a little list here, that's the phospholipid arranged in a polar region. Okay, that means they love water, and the hydrophobic lipid fatty acid that doesn't like water. So as we know, water and fat don't mix, right? So it's the same thing with this membrane that the cell has. So here it is here. These are our phospholipid bilayers, right? So it's two layers. The outer layer has a polar head, and the inner layer has a polar head, and they have hydrophobic tails, right? These are the fatty acid parts of the phospholipid, the lipid part, okay? Didn't know lipids were made of fatty acids. And it's semi-permeable, which means it's not a wall, okay? It does have ways to get in and out. So here's our gram-positive, here's our peptidoglycan layer that we're staining today, and here's our plasma membrane. The plasma membranes of the gram-negative and the gram-positive. The inside looks pretty much the same, except the gram-negative has an outer membrane of this phospholipid bilayer, okay? It has a real thin peptidoglycan layer, and then the gram-negative has what? It can have a capsule out here, correct? So this is very important that you know that the gram-negatives have two membranes covering them. And then we have both have plasma membranes that protect the nuclear inside of the cell. Peptidoglycan big layer here for gram-positives, small for gram-negative, and then an outer membrane for the gram-negative. So we just talked about how it's permeable, it's selective permeable, so things that are based on their size and charge might not get in. So big things won't get in, but small things can. Positively charged things might not get in, okay, but a negative might get in. Nutrients pass in and out of the cell, passive transport. We have active transport where we got to use energy to get things in and out, okay, to cross the membrane. We got to have some help with some carrier enzymes called permeases to permeate. The enzyme involved in energy production is also located in this membrane, and those that remember what we're about to get to, which is physiology, right? The electron transport mechanism that produces most of our ATP is located where? Anybody remember? We have the glycolysis where we take sugar and break it apart into pyruvate, right? And pyruvate enters into the Krebs citric acid cycle a couple of times and gives us some energy, but all those energy molecules go where? They head out to the membrane where there's electron transport in the membrane, and we get hydrogen going one way and any NAD going the other way. Do you remember any of this? anything about this at all? Okay. So how much, let's put it this way. How much of intro to micro did they spend on metabolism and physiology? You all remember?
Maybe when it's
A whole chapter, maybe?
maybe.
Maybe? Or did they skip that part? Say you got that in biology, let's move on.
A little bit.
Okay, good. All right. Well, this part of this electron transport system that happens at the cytoplasmic membrane is where the oxidative phosphorylation takes place. How we doing, Lauren, Rachel, Brandy? Did you ever
Good.
get a hold of Kathy?
No, I didn't.
No, sorry. We were doing so good. We had her on every call so far. All right. So the cytoplasmic membrane releases hydrolytic enzymes. Okay. Pathogenic bacteria, what we're interested in here, such as Corynebacterium diphtheria, is the causative agents of diphtheria, produce virulence factors in the form of exotoxins. So what happens with the diphtheria is that exotoxins are produced, and that's what makes you sick. The cell membrane is involved in the biosynthesis of some lipids, proteins, polysaccharides, or sugars, carrier molecules. The cell membrane is also the site of DNA synthesis and endospore formation. So there's some more about the cytoplasmic membrane for us to revisit later, okay? So this is kind of our intro to the membrane. All right, switch gears now to the appendages. Now this you probably have already had before. Shouldn't be too hard. For the quick review here, that we know we have bacteria do have flagella. They can, okay, so the whip-like appendage helps with locomotion. So you might want to know how a bacteria moves around, right? They might have a flagella for motility. But that flagella is very antigenic, which means it's going to elicit an immune response. And we can identify this flagella, especially true with the salmonella. And then one of the things we're going to learn is what is the antigen called that is associated with the flagella, and it's called the H antigen. And we're going to revisit this later when we get to salmonella. But if you have a flagella, the antigen that we call that is the H antigen, and we can serotype that GI pathogen. So we're going to have certain antigens produced by what we're looking at. So if the flagella is detected, we'll have what's known as an H antigen. We have different types of flagella. We have a monotrichous, which is possessing a singular flagella, A here. We have a locotrichous, which possesses a tuft at the flagella at one pole. We have an amphitrichous, like ambidextrous, I guess the way you can look at it. We have a left and a right. And we have a periotrichous, which just has flagella around the entire surface. So a lot of hair on this one, just everywhere. Okay, so those are some names for our flagella. We have smaller projections known as pili, and they're found on gram-negative bacteria. So you'll have a chance, I don't know if you'll see the pili today, but you'll have a chance to gram stain. They're probably a little too small for a 1,000-time light microscope. When we see Neisseria gonorrhea and other gram-negative bacteria require the pili to attach to the cells of the genital urinary tract. And without these pili, they cannot cause disease. So if we have a way of knocking it out, or a pathogenic only version would be with the pili. There's also something called the sex pili that are imported in what bacteria do, which is conjugation. Okay, so they attach to each other, and it's hollowed core so they can transfer DNA from a donor bacterium to a recipient. Didn't know if you knew that bacteria could do that, but they do. And then our axil filaments, or our filament structures, we have these that are made of protein that provide motility for the spirochetes. So we have flagella, pili, and axial filaments. They're within the periplasmic space, and they allow the spirochete to move. About the only thing I want to point out here with bacterial cytoplasma is this is where inclusions are. The amino acids, sugar fatty acids are found inside the bacterial cytoplasm. The cytoplasm, of course, is that inner space of the cell within the cytoplasmic membrane, right? So now we're inside that. So inside there we have ribosomes, and if you don't know much about ribosomes, that's how we get proteins. Proteins are synthesized from ribosomes, because ribosomes are going to do what? They're going to read the DNA or RNA, right? Really, what do they read? They read messenger RNA, right? To form a protein. So these 70S, and I know this doesn't make a whole lot of sense, but the protein synthesis in a bacterial ribosome is known as the 70S. And I know 50 and 30 don't add up to 70, but that's correct, okay? But it's just the size. Okay, so it's two subunits come together to form the 70S ribosome. Okay, so the 80 with us, our eukaryotic ribosomes, are 80S. And they're composed of 60 and 40. I know that doesn't add up to 80, but it's not supposed to. It's the size. Okay, so the 60S subunit combined with 40S, it makes the 80. And the 70S is the 50 and 30. And you're like, I'm never going to remember that because that's going to be confusing. But hopefully you will, that 70S is our bacterial ribosome size. So this is that cytoplasm we were talking about. And then we also have our nuclear membrane, or there is no nuclear membrane. I'm looking at my nucleus. There is no nuclear membrane like eukaryotes, okay? So in our cells, we have a nuclear membrane, that this is where the RNA comes out of, right? The RNA comes out of the nuclear membrane through the nuclear pores, and you get what? You get synthesis of the proteins out in the cytoplasm. So a bacterium doesn't have that nuclear membrane. So the bacterial DNA is arranged in a long, narrow, circular double helix, okay? So this is the DNA of the bacteria. If you look, you see something over here called plasmid, right? Plasmid's like a little piece of genetic material. So it's an extra chromosomal DNA that serves two functions. Plasmids serve one function, which is they're a role in toxin production, especially with the Corneobacterium diphtherium. And it's transferred by a prophage, which acts as a carrier mechanism. So we're not going to get much into that, but plasmids they're virulence factors too. Okay, so if I can be transferred right out, and then I become an exotoxin, then I am a virulence factor. I cause a more damaging disease than if I wasn't around. The other thing that's really important that a plasmid does, antibiotic resistance is transferred from one bacterium to another through the transfer of the plasmid. Remember that sex pili that we talked about earlier? I said they could connect a hollow bore, and that plasmid can be what? Transferred over to the other bacterium, which is intaked into the genetic material of that one. So when you say, well, how in the world did that bacteria become resistant? It never saw that antibiotic. But the bacteria that saw that antibiotic and formed a resistance to it transfers it to the other one. So then they both can resist your antibiotic, okay? And that's how antibiotic resistance occurs. And that's what we're going to spend time on. A little bit past midterm. But I had a really good thought. that last time we did this class last fall, we waited till that time after midterm to put antibiotic resistance and antibiotics into the conversation. I didn't think it really worked well. So the thought I had over the last couple of weeks was, I think what I want to do is when we get into the specifics of the organism, we want to somehow find the space to put the antibiotic for that class in there, to bring it up, at least start to introduce it as we work through it. And then when we get to the end, when I say, hey, you need to know all these antibiotics, and you're like, well, what do they even do? And then, oh, you're supposed to remember from Staphylococcus that one was part of that. So we're going to do an effort to bring those antibiotics, because that is a big part of your microbe, is to understand and be helpful in that. I talked about patient care team. It's your ability to go forward and say, hey, we've got this organism and we're seeing every patient at this hospital has a resistance to this antibiotic. We must be using this one too much, right? So usage and effect can be changed if you're able to give some little helpful hints to the physician and say, here's our monthly report from the micro lab, and these are the problems we're seeing. We're seeing a definitely increase in resistance to this antibiotic. It must be your go-to. And some doctors are like that. They go to a go-to over and over and over and over. And then all of a sudden, all the patients start getting a resistance to that go-to, and they end up getting sick at the end. Anyway, that story we're going to start telling more and more as we work our way through the chapters. So if this seems foreign to you right now, and you're like, I don't want to wait till after the midterm to bring it up, then I'm going to bring it up more and more inside the lectures. Okay, so where are we on our little notes? Are we getting close to the end? We are, aren't we? We doing good? Okay, now keep me on time. Don't let me just spend all right. I've got 12:07. What time does this class end? 12:15. Okay. All right. We got eight minutes. 8 minutes. All right. The purpose of I'm not going to rush. We'll get where we get, But here's the metabolism part. I told you earlier, about the Krebs citric acid cycle. We can talk about catabolism and anabolism. Catabolism is breaking things apart. Anabolism is building things up or synthesizing organic compounds. I taught that in phlebotomy class years ago. So I think you should have an idea. It's in A&P, right? Physiology, right? Part of A&P. Y'all do physiology in A&P, right? So here's glycolysis, right? Taking glucose to glucose 6-phosphate. Remember, this is just lovely. It takes an energy source here at ATP. That gives us to fructose 6-phosphate. We take another energy source of ATP, so it's costing us energy to run glucose. This is breakdown of glucose, okay? So you kind of get the idea. And if I was giving my micro class, if I was doing a general micro class, I got a big sheet of paper during my exam, and I had to fill these in. Did y'all have to do that in Intro to Micro? Did y'all fill in these structures and names as you walk through this? Yeah, I think it's become like old hat. Anyway.
You had to do a name or two.
A name or two, but you didn't just have to memorize this. Okay. Do you understand how phosphate groups work? Do you know that's a phosphate group when it's PO3 right there? Right? Those are key, because what we're going to do there is we're going to use that phosphate to move it around, change the name, and what you're seeing as these NADHs are coming off. So we cost us with glycolysis, it cost us 2 ATPs to produce 2 ATPs, but we also got NADH, and remember it's glucose. So when it gets to pyruvate, it's going to be 2 pyruvates down here. And those 2 pyruvates enter the Krebs citric acid cycle at that point.
I think I like you. That was chemistry.
That was chemistry here?
Straight, yeah.
Okay. Biochemistry, maybe, yeah, okay, all right. DNA replication, you know DNA opens up. You know that we have to do polymerase. I'm not going to spend a lot of time there. Chapter 2 does really well going over this. Here's protein synthesis with our messenger RNA. This shows you how it works, right? Looks like a eukaryotic cell, which we were talking about, the nuclear pores here leaving and making this protein right here. So this would be what? The 80S ribosome, since it's eukaryotic. And then we have these amino acid codons that come in and move and bring the amino acids in, and then they make this chain of polypeptides, right? And then you end up with a protein at the end. And then lastly, our bacterial growth, which we'll be looking at, is it has different. The way we grow is lag, log, stationary, and depth. And it's basically this. Okay. I think we finished. Did we finish on time? 5 minutes, right, left for questions? Okay, so the main thing is to get an idea of what we're going to be doing downstairs. So we have the, where we can definitely have what else here, got the chat here. This one didn't work, okay. We will share screen. We'll go back just to the camera. Okay, so gram staining today. We'll start in about 20 minutes downstairs. Remember, you come in one door, you get your temperature checked. There shouldn't be any lab next door, so it's just you today on Tuesdays and Thursdays. There will eventually be some Heme 2 students come in and do some slides next door to us. But we will exit the other door. Miss Carly Farmer will be there to help us get started. Get your tables and we'll get our stuff out. And it's no rush. And those that were in immunology yesterday, section 1, okay, is always the experimental section. Okay, so we'll get a feel for where stuff is. Do we have everything out? And then everybody in section 2 and 3 gets a benefit from that. And I apologize, but 2 and 3 went so much better than 1. 1 didn't go too bad, but we had crystal violet too thick. We had 9 tubes instead of 4 tubes. We had some typos that we didn't cover, all that good stuff. That's the first group. We think we're better today. We're going to have plates out already grown. We're going to get some more plates out for streaking. We'll have we're trying to put a gram stain station at every table so you don't have to get up and share one. The gram stain is not going to be, swish, swish, swish. It's going to be flood and count, right? So if you need you should have a clock. If you don't have a clock, it's a 1 minute and you watch the clock, big clock on the wall. And that's just a random time. It just works at 1, 15 seconds and 30. And it's just how but as you go have the procedure downstairs, then once that's all done and you've looked at it on the scope, then we're going to move to the streak for isolation to finish up. Okay, so you're going to streak with, I know the picture shows a flame. We're not going to flame up anything down there. We use sterile plastic loops in micro. So those will go in the biohazard at the end when you streak with them. You are working with real organisms, okay? So cell phones need to be put away, lab coats need to be on, gloves need to be worn, okay? Don't have those cell phones. I saw some yesterday in pockets and kind of hanging around the tables. They're not to be out and about, okay? And then if you leave for any reason, you need to not only take your gloves, but your mask off too. Change mask as you leave to go to the bathroom, because that's just like wearing your gloves out to the bathroom. We don't do that. So the lab coat and the mask can be left, right? Reapplied once you get back, but no cloth mask. And I think we did pretty good yesterday, but you are working with organisms that are alive today. So these are not pretend, they're not non-pathogenic, okay? So we're giving you real organisms, and I think you did that in intro. I think you really worked with real stuff, but we have everything, okay? So we brought out some some obvious ones that you'll be seeing later. And then we definitely want you to streak for isolation with these and see what they look like on Tuesday. Any questions? Nothing at all. All right. We'll see you downstairs in about 15, and we'll get started. Thank you, girl, Rachel, Brandy, and Lauren. And I'll send Kathy something to let her know. And we'll see what this recording does, and I'll get it posted.